Hello everyone, welcome. We're going to be looking at renal system regulation of changes in hydrogen H+, and what we're going to look at there is how that regulation occurs in contributing to a stable pH within the body. So either accepting or donating this hydrogen. So acids will typically free up those H pluses, making the body and the blood more acidic, and then bases such as HCO3 minus in the blood will be accepting those protons to make it more those extra hydrogen ions to make it more basic, and the body has to keep a specific pH within the blood itself and the body so that all the wonderful metabolic functions can occur. So we'll look at how that occurs within one of the lines of defense, which is the renal system, and we'll go from there. So we'll look at the basics of the process, the importance of how this works, and some recent research specifically dealing with organ transplantation and its importance with that. So before we get started on this, let's talk about the lines of defense that we have for keeping the body with a stable pH. And the first line of defense are a bunch of chemical buffers within the blood and the body, and those are the first line of defense. The second area that we have is with a system we've previously discussed, and that is the respiratory system with the exchange of oxygen and CO2. That is another line of defense. And then we're going to get into very specifically the third line of defense, which is within the kidneys, specifically the renal area itself, and how that plays a part in keeping the body balanced with its pH. The pH is anywhere between 6.8 and 8.0, usually about a 7.4 is what we're striving for with that sweet spot there. So within the kidneys, we're looking at two areas very close to one another. We're going to look at the proximal tubule and the distal tubule and the collecting ducts. So there are two different types of cells that we're dealing with, and the basic process we're looking at first in the proximal tubule. We have H+ that is secreted primarily and secondarily through active transporters with an ATPase pump, and these pumps are exchanging sodium and hydrogen ions, and they're secreting that, the hydrogen for sure, and then they're reabsorbing the sodium. And as this is happening, those hydrogen ions that are mixed would then combine with the HCO3 and effectively make them neutral. So the more hydrogen ions that we have, the more acidic the blood is going to be. The more HCO3 minus we have, the more basic the blood is going to be. And so within the proximal tubule, we have these cells that we see right here that are then helping keep things balanced here in the proximal tubule. Here's another example of that and how that is going from proximal tubule and back to the blood to keep the blood at a very specific pH. So now we're going to look at the distal tubule, and the distal tubule has a different type of cell. These are in the collecting tubules as well. They have intercalated cells, and you have two types. You have a type A, which secretes the hydrogen ions and has HCO3 minus and potassium that it reabsorbs, and again using the same pumps as discussed before, hydrogen ions and ATPase pumps, and it's also using hydrogen and potassium ions as a pump. And we also have another type which are called the type B, and these are HCO3 minus secreting and H+ reabsorbing and potassium secreting. So the type A, as again you can see, they have a lumen and they're located on one side here. lumen here we have the H plus pump on one side, and the type B has the HCO3 in the opposite, located on the other side. So as we're shuttling these things in and out, as we're secreting this, we're either trying to balance the amount of HCO3, or if we're secreting this, we're trying to balance the amount of H plus that is going on. And this is all happening within the distal tubule, which is then next to the collecting duct, and this collecting duct is either going to send the excess ions out of the body through the urine, or it's going to have them be reabsorbed so that we can keep this wonderful pH balance that we are talking about. And that is the basics of how this is going to work. I don't want to get too technical because the audiences that we usually deal with want the basics so they can understand this. Normally we deal with acids and base in a little bit more complex. These are our acids. Anytime we have an extra hydrogen ion, that's going to make the blood more acidic, and anytime we have anything that has a negative ion, in this case this carbonate, it's going to make the blood more basic. And as mentioned before, there is a sweet spot that we're looking for with the body. So the importance of this is that metabolic activities must occur between pH values of 6.8 and 8.0, which is, very low on the acidity and on the slightly basic. If we think of how the pH scale works, usually 7's a neutral, and 6 tends to be a little bit more on the acidic side, 8 being more on the basic side. We're looking at anywhere from 7.2 to 7.4 for normal pH. If the pH of the body, again, if it goes above 8 or below 6.8, the normal metabolic activities that we have don't normally occur. There's too much acidity breaking things down, or it's too basic. We're destroying the basic components of the cell, unraveling and denaturing the proteins and things that are actually holding the cell together. So we cannot have that done. As those proteins, of course, start to unravel, the cells don't have any cohesiveness. If the cells don't have cohesiveness, that, of course, has a cascading effect to the tissues, which then goes to the organ and to the body itself. So very important that we keep those pH values there. And as the kidneys are filtering the blood with those chemical buffers, it makes sense that they would be another line of defense, keeping the body within homeostasis and giving it the necessary requirements to carry on these metabolic activities, because without these metabolic activities, the body essentially dies within minutes. So very important as to how this works. Now let's look at some of this recent research that we have so we can get to how interesting that is and how important that is as well. So when we're looking at transplanting organs, one of the more common organs transplanted are kidneys, because with kidney failure, you're looking at the way the body can't filter blood or liquid waste that it has, and then can't send that on to be expelled or retain the water and so forth that is necessary for the body to continue to function properly. So one thing that was looked at is some kidney transplantation, and there is a particular solution that was called a perfusion solution, and it's looking at maintaining the renal pH and preserving kidneys for transplantation. So as we're looking at preserving these organs for those folks that are organ recipients, we need to make sure that they stay in good function and that they're going to operate well when they're transplanted from one person to the next. So in order to do that, they're looking at this perfusion solution, which maintains a good renal pH, keeping that so that the organ is in homeostasis, and that the body, as the new body is being prepared for that, they put the organ in the body, that it will not reject it and cause more complications and problems. Of course, with every organ, you need to keep them cold, and one of the things they looked at is putting those organs through a certain hypothermia. Again, anytime you change the temperature, you may be also affecting some of the solution, that its cohesiveness and how it functions. So this perfusion solution that they used showed that it was very effective in keeping the pH at an optimum level, so that organ recipients would then have a functional kidney for the rest of their lives. So we have that information going on. I hope you've enjoyed this presentation, and here are the references that we have, giving credit where credit is due for the images that we have, as well as the studies that have been done, and the fascinating things that we learned there, and the importance of maintaining its pH within the body. So thank you for joining me, and we'll try and catch you at another time.